Welcome each and every one of us to this lecture. We are glad to have all of us here. My name is Anyango Esther. I'm a pharmacist by profession based in Nairobi, Kenya, and one of the members of the Africa Pharmaceutical Network. Glad to be joined by all of you and looking forward to this lecture. So, as a form of introduction, for those who may not be too conversant with the African Pharmaceutical Network, I will just give a very brief summary of who we are and why you should join us. So, the African Pharmaceutical Network is basically just a network that was founded to bring together all pharmacy stakeholders, professionals, for the purpose of sharing knowledge, capacity building, really, and our end goal is to improve reliable access to safe, efficacious, and affordable quality medicines. So that's just basically who we are. We have a website, and I will post our link on the chat box just to allow anyone who may be curious, who would want to know more about us to check us out. We do have a membership. And of course, there are so many benefits that comes if you are a member. Not only do you network with like-minded professionals, but you'll also have access to trainings, access to forums to allow us really just to gather together as pharmacy professionals for the purpose of sharpening our skills and building that strong community of professionals here in Africa. We are delighted to be hosting this webinar today. We also have the Pharmacy Academy. Those who are in pharmacy groups, at least here in Kenya, have had the privilege of accessing or receiving marketing for our Pharmacy Academy, which is basically the trainings that we offer as African Pharmaceutical Network. So among the trainings that we offer are the regulatory affairs trainings. We also offer trainings in supply chain and offer trainings in pharmacovigilance and patient safety. So we invite you, if you're interested in upgrading your skills in any of those three areas, to kindly reach out to us. We are accredited by PPB. We are affiliated by with FIP, International Pharmaceutical Federation. So I think if you're wondering if we are skilled enough, just to get it out there, that we are skilled enough and would be happy to offer our trainings to you, of course, at a fee, but at an affordable fee. So that's just about our Pharmacy Academy. Our lecture today, our webinar today, is entitled Understanding Regulatory Requirements for Bioequivalence Studies in Kenya. So for those of you who are in the pharmacy space, you know that BE studies is a heavy topic, and a topic that I think as Africa we need to upskill ourselves and really just get us understanding about this topic. Our speaker is Dr. Felicitas Chepwogen. She's the head of Bioequivalence, the Bioequivalence Division at the Pharmacy and Poisons Board in Kenya. The Pharmacy and Poisons Board, for those who are not conversant with it, is basically the national regulatory authority here in Kenya. So a brief about Dr. Felicitas, and thank you so much, Dr. Felicitas, for agreeing to facilitate or to present in this webinar this evening here in Kenya. She is a dedicated pharmacist with a deep commitment to pharmaceutical regulation and harmonization efforts in the East African region. She has a career that spans over a decade at the Kenyan Regulatory Authority, the Pharmacy and Poisons Board, and has played very many roles in pharmacy practice. She, as an area to note, she spearheaded the development of minimum requirements for the pharmacy student training and pharmacist registration in Kenya. So I think many of us pharmacists owe a lot to some of the work that Dr. Felicitas took part in. Her expertise extends to fostering relationships with local manufacturers, championing the harmonization and implementation of regional medicines registration and regulation policies within the EAC, the East African Community. She also played a role in Kenya's ratification of the treaty for the establishment of the African Medicines Agency, also a really big thing here in Africa. And of course, this just marked the country's commitment to regional healthcare integration. Currently, as I had already mentioned, she serves as the head of the Bioequivalence Division at the Pharmacy and Poisons Board and continues to contribute significantly to the advancement of pharmaceutical standards and practices. She holds a Bachelor of Pharmacy degree from the University of Nairobi and has furthered her education with a Master's of Science in Tropical and Infectious Diseases from the same institution, and has also a postgraduate diploma in project management from the Catholic University of Africa. She embodies a blend of practical experience, academic excellence, and a passion for enhancing healthcare systems across borders. It's a delight to have you, Dr. Felicitas, and I know you'll be joined by your colleague at a later stage, Dr. Brian, and I think at that time we will be able to give him an introduction. I will pause here and give you the mic and allow you to deliver your presentation. In case you have any questions, please feel free to use the Q&A tab for the webinar. In case you have any questions, any comments, please feel free to use the Q&A tab or the chat box. All right, so I think that's it from me. So Dr. Felicitas, you're welcome for your presentation. Thank you.
Thank you, Esther, for the invitation. And also thank you to the African Pharmaceutical Network. I listened to one of your webinars some time back. I didn't know I'll be the next to present. I've benefited from your network. I've understood so many things about even harmonization. And today I would like to share my experience or the knowledge I have about bioequivalence. Indeed, there's been a journey. We have been on and off when it comes to the requirements of bioequivalence in Kenya for locally manufactured products. And this process started in 2010 when we implemented the CTD format of dossier submission. And it was a requirement that we have all the 5 modules submitted. But due to lack of capacity at the national level, we exempted the local manufacturers for some time. And now we are undergoing what is called WHO global benchmarking assessment. And one of the requirements is to have the same criteria, either for local or foreign products. We need them to have the same treatment at Pharmacy Board, and that is why we've started this agenda again of mandatory bioequivalence for all products. We have done a number of we have achieved a number of milestones, and we adopted the EAC guidelines. It's called a Compendium of Medicines Evaluation and Registration in 2015. One of the key template or guideline was the one for bioequivalence. So we've been having these documents in place, but I know we have been also evaluating bioequivalence at the Drug Registration Department of Pharmacy Board. And it only came apparent last year that we need to be a bit rigorous, because when we were assessed by WHO, they made an observation that we are not applying the same criteria. So that is why we are here. I would like to introduce my colleague, Dr. Brian Kariuki. Is one of the staff at the Pharmacy and Poisons Board designated to the Bioequivalence Department. The nitty-gritties of what we receive, the applications we receive, and what we check for during the assessment will be done by Dr. Kariuki himself. The questions will be directed to me and also Dr. Brian Kariuki. So I'll do the presentation. As there's been mentioned to you, I've been in the regulatory environment from 2006. I've worked for the East African Community for 4 years as a full staff, and also I've been the focal person for the EAC since 2013. I also facilitated the ratification of AMA Treaty in Kenya. And for me, there's been a good experience. I've interacted with many people. And through this interaction, I've learned a lot. And I'm not saying I'm perfect. I'm still learning. And even when I'm presenting here, I know there are people who are better than me in bioequivalence. So if there is anything clarification which might be beyond my knowledge, I will also welcome those in the group, the attendees, to chip in. Please, let's now start the presentation. The first slide. This is the outline of my presentation. I will not discuss so much on this, but at least you will be guided by the history of bioequivalence, the legal framework at the Pharmacy and Poisons Board, the different designs and the study subjects, the parameters to be assessed, quantification and validation of analytical method, and also the topic of biowaiver. Next slide. The history of bioequivalence. Bioequivalence is not new topic to all of us, but at least we know the Hatchman-Waxman Act of 1984, where the Food Drugs Authority required only bioavailability for generics. In US, it's called abbreviated new drug applications. And in 1995, we had the GL amidon requirements. The WHO came in 1996 with FDA BCS guidelines, which was developed in 2000. The WHO TRS 937 was developed in 2006, all speaking to bioequivalence. There have been improvements with even AU, BA, BE guidelines. And in 2010, FDA updated their guidelines. There is a WHO TRS 1992 in 2015. And now the latest guideline is WHO on bioequivalence, WHO guidelines on bioequivalence TRS 1003 Annex 6. And following the harmonization initiative, we have been having convergence of regulatory requirements and the need for biowaivers for class 1 and 3. Next slide. The legal framework. The Pharmacy and Poisons Board, the previous slide, please. Yes, the Pharmacy and Poisons Board is mandated under CAP 244, that is version of 2019, Section 3B 2P, to ensure that all medicinal products manufactured, imported, or exported from the country conform to the prescribed standards of quality, safety, and efficacy. And Section 3B2O approve the use of any unregistered medicine substance for purposes of clinical trials and compassionate use. And Subsection P, approve and regulate clinical trials on medicinal substance. We developed the 2022 rules in the year 2022, and these rules were made to guide the conduct of clinical trials in Kenya, but Section E of these rules provided for the establishment of bioequivalence for registration of generic health products. So from these rules, investors or government agencies can come up with clinical research organization, or what we call bioequivalent centers. And also it gives pharmacy board mandate also to regulate bioequivalence studies in the country. Next slide. What is bioequivalence? Bioequivalence is a comparative clinical trial study, and it is targeted for generic products which are required to conform to the same standards of quality, efficacy, and safety required for the originators or innovator products. So we can declare that two products are bioequivalent if they are pharmaceutical equivalent or pharmaceutical alternatives, and there are bioavailabilities in terms of the parameters we are going to discuss, the Cmax, Tmax, and AUC, after administration of the same molar dose under the same conditions are similar to such a degree that the effects can be expected to be essentially the same. When you look at the diagram on your right side, there is a dissolution profile or bioequivalence profile of two molecules, formulation A and B. A is the comparator, B is the test product. When you look at the concentration time profiles, you can see they're almost aligned. And what we are looking for is that in bioequivalence, there should not be any significant difference between the two formulations, so that we are able to say the products are interchangeable and the clinicians can prescribe either the generic or the original product, that is the innovator. The next slide. So we have 4 types of bioequivalence studies, and the sensitivity varies. From pharmacokinetic studies, the sensitivity is high, and as we go to in vitro studies, the sensitivity becomes lower. So the first one, where we measure the concentration of the API in the body fluids like the blood, serum, plasma, and also urine, the sensitivity to detect the difference is high. We have pharmacodynamic, which measures the pharmacological effect. Comparative clinical trials check the clinical endpoints. In vitro studies now is when we do dissolution studies. They are not normally recommended as surrogates for bioequivalence, but if the method is validated, they can be used as a surrogate for bioequivalence, and it also provide information in case we have biopharmaceutical classification bioeval. There is in vitro, in vivo correlation, which we'll talk more about this. But in vitro, in vitro correlation is a mathematical formula which tries to compare the results of in vitro and in vivo for any particular drug. And it guides to show whether we have any similarities in their actions on therapeutic effect. Maybe you continue clicking, because it's a diagram which shows what do we need when you are submitting an application for generic product registration. We have the innovator on your left side and the generic product on your right side. All the information required are the same, but preclinical studies are not necessary for generic products. Please continue clicking. What is important for generic product application is bioequivalence. That is efficacy and safety studies. And that is what has been the missing link in some of the applications we receive at the Pharmacy and Process Board. So as you can see from this diagram, all the information required are the same, but normally when you get a dossier for generic products, they say preclinical studies are not available because this is a generic product. But what is more important is, as I have said, is bioequivalence studies, that is module 5. And That is what you are going to discuss. That is the topic of discussion today. What are the requirements for generic medicine? A generic medicine must contain the same API as the innovator drug, that is number one. It should be identical in strength, dosage form, and route of administration. Should have the same indication, be bioequivalence, and must meet the same batch requirements for identity, strength, purity, and quality, and should be manufactured under the same standards of good manufacturing practices. Where is pharma? Sorry, I don't know why I'm calling bioequivalence pharma. Bioequivalence required. It is required in vivo documentation for equivalence when there is a risk that possible differences in bioavailability may result in therapeutic equivalence. When the innovator develops a new formulation, and especially during drug development processes before approval or as a variations of the market product after approval. I know we've been receiving a number of variation applications. Some require in vitro dissolution as a surrogate for bioequivalence, but some requires a full bioequivalence study. And I know queries have been raised on the same, and my colleagues, some of them are here. Dr. Brian will represent them and will give us more information on the same. When the innovator also develops a new dosage form, like line extension, and when a multi-source generic product is applied for marketing authorization. We can continue. So there are 4 experimental designs for bioequivalence. I'll say something about each of them, the advantages and disadvantages. The first one is single dose crossover study, parallel design, multiple dose crossover, and replicate design. All these are the designs used in bioequivalence, but they differ in complexity and to a great extent determine the costing of bioequivalence. Single dose crossover design, as we'll see later, you can have a number of 12 subjects, but when it comes to parallel design, you require a lot of subjects because there is no crossover, and it has an impact on the costing of bioequivalence studies. Next slide. So the first one, the single dose crossover study, because subjects are divided into two groups, group A and B. No, okay, group 1 and group 2. Treatment A and B, there are 2 periods, 2 sequences, and there is randomization of the subject in the first case, and then an allowance of a washout period. There is efficient use of subjects, and the subject itself, himself or herself, becomes a control of itself. Like Felicitas can take treatment A and then take treatment B, and I can be able to see which treatment is better than the other one, or there is no significant difference among the two products. There is also control of inter-subject variability. The disadvantage of this one is the carryover effects. Yes, we say the washout period is 5 times the half-life, or maximum 7 days, but there is a potential for carryover effect and might influence the response to treatment B. It's not suitable for drugs with long half-lives, and that is why we have the parallel design coming next. Example of a single dose crossover study is comparing generic and reference version of a single dose oral antibiotic like amoxicillin. We can go to the next design. The next design is parallel design, where we have a randomization of participants. You administer treatment to one group, no crossover, and the another treatment to sequence 2. And in this case, it is common with long half-life products, which can take up to many days. So we are sure with this one, there is no carryover effect because a subject only takes one treatment and is suitable for drugs with long half-life. This one, the disadvantage is increased subject variability and requires larger samples, which we have said will also be costly for the sponsor. And then now, Example is comparing pK of different formulation of sulfadoxine and pyrimethamine, fixed dose combination tablet sulfadoxine has a half-life of 7 to 9 days. Next one, multiple dose crossover effect. Evaluate the steady state pharmacokinetics and increase sensitivity to the difference in formulations. This one now require longer duration, and there is potential for carryover effects because you are giving patient different treatments for a longer time. You keep changing from product 1 to product 2, product 1 to product 2. Example of this is comparing generic and reference version of a multiple dose oral antidepressant such as sertraline. Next slide. The replicate design now provides, it could be the best because it provides robustness to results. We have subjects which are randomized to sequence 1, 2 sequences, and then the periods, we have like 4 periods. We have period 1, period 2, period 3, and period 4. And you administer test, reference. This one has an increased cost because of the number of the duration you are going to administer the products, and also a case study on the small pressing and nasal pressing nasal spray preparation is an example we can give for this one. As I said, there are 4 types of equivalence studies. We have done the first one, pharmacokinetic. The second one is therapeutic equivalence with clinical or pharmacodynamic endpoints. These are for products which have systemic action but no measurable concentration in plasma. An example of it is products for local action, except solution with same composition, pharmacodynamic designs for cutaneous corticosteroids, and also pharmacodynamic design for bronchodilators. Clinical variables required in other cases. There is no FDA model for inhaled corticosteroids, and dermatopharmacokinetics and dermal microdialysis for cutaneous, and also in vitro acceptable in very few cases. So bioequivalence products could be considered therapeutic equivalent with clinical endpoints if such a study were performed. The bioequivalence confidence intervals of 90% for the AUC and Cmax, and the range is between 80 to 125. If the parameters are outside 80 to 125, they are not bioequivalence, they are called non-bioequivalence. And bioinequivalent is when confidence intervals, the 90% confidence intervals is completely outside the 80 to 125% range. Therapeutic equivalence can be concluded between 2 immediate release and prolonged release formulation. And therapeutic equivalence can be concluded with different drug substances like omeprazole and lansoprazole. So we cannot assume that 2 products are bioequivalent, and that is why we need bioequivalence. And the reason is we can have differences in the raw materials. And the raw materials, like for a drug, they can have different particle size, different polymorphs, and also excipients, because we have different grades of excipients. I know we do control the source of our excipients, but sometimes you can have different grades and it will affect the bioequivalence or bioavailability of your products. The manufacturing process, dry versus wet granulation, the equipment also can influence bioavailability, site of manufacturing, and batch size. may affect the bioavailability of pharmaceutical equivalents or pharmaceutical alternatives. So bioequivalence has to be demonstrated, and the possible differences are listed there. And that is why we need documentation of bioequivalence between the test and the reference product. The study subject. As I said, it varies depending on the study design, but a minimum of 12 subjects is required. What determines the number of subjects? The first one is the error variance. We require the coefficient of variation associated with the primary parameters. This is estimated from a pilot experiment or from previous studies which are available in the literature. The level of significance we desire in most cases is 5%. The statistical power decide, and the mean deviation from the comparator product compatible with bioequivalence and safety and efficacy. And also the confidence intervals and the range, which is acceptable limits of bioequivalence 80 to 125 for log transform data. When you are doing statistical analysis, exclusion of data for statistical or pharmacokinetic reasons alone is not acceptable. Retesting of subjects is also not recommended, and potential reason for exclusion should be included in the protocol. Next slide. So I've said we have two products. One is the test product, and the other one is the comparator product. For biophytal BE studies, the test product should meet two criteria. The production patch should provide a high level of assurance that the product and processes should be feasible on a commercial scale. And the test product should usually originate from a batch of 1 out of 10 or 100,000 units, whichever is greater, unless otherwise has justified. So unless otherwise justified, the asset content of the batch between the product and the reference product, the difference should not be more than 5%. The reference batch throughout the product life cycle should be sufficient. That is what we have said, more than 100%, 100,000 units, and should be identical to production batches. Adequate documentation generated and maintained for the reference batch. I know at some point you'll ask me, where are we getting the reference product for our studies? The critical quality attributes also for the reference products in terms of the particle size, polymorphism, solubility should be documented. And also the CQA attributes of the finished products in terms of selection and proportion of excipients, discriminative dissolution should be provided. And the basis for control parameters, that is dissolution specification. The study should be done at the 3 physiological pHs of 1.2 acidic, 4.5, and 6.8. So for every bioequivalence study, you need to conduct dissolution in vitro dissolution at the 3 physiological pHs. For test product, which one are we going to use? We'll discuss later the issue of bioeval. If there is more than one strength to be used in B studies, the higher strength is normally chosen, and this is for linear pharmacokinetics. Higher strength is also used for drugs with non-linear pharmacokinetics. If the concentration time profiles is greater than proportional increase in AUC with increasing dose. The lower strength will be used in linear range for drugs with non-linear pharmacokinetic with saturable absorption and resulting in less than proportional increase in AUC with increasing dose. And the higher strength and lower strength is required for non-linear pharmacokinetics with products with limited solubility, resulting in less than proportional increase in AUC with increasing dose. Formulation should be directly proportional and manufactured using the same manufacturing process. The comparator product is normally the product which received the first marketing authorization, that is the innovator product. We have the comparator product is normally interchanged with reference listed products depending on the region. So for ICS region, they use reference listed drug, which is if innovator is no longer available. The Pharmacy and Poisons Board, any regulatory authority should be able to provide directions if any of the above guidelines is not sufficient, like in getting the reference product, and also the need for harmonization of these reference products within the region or continentally. So if the innovator product is no longer marketed in the jurisdiction we are in, the selection principle is prescribed by the WHO, and this is TRS 937 Annex 8. The link for comparator products by WHO for pre-qualified products is provided in the slide. Study conduct. When you are conducting your study, you need to ascertain the potency and in vitro dissolution characteristics or multi-source drug products and comparator products. The contents of the API should be close to the label claim, and difference between the test and comparator or the reference should not be more than 5%. Otherwise, you need to do what is called potency correction. We have said you should always select the highest strength, highest marketed strength. A higher dose, more than one dosage unit, might provide analytical difficulties. But you can also choose a lower strength for safety reasons or for APIs which are highly soluble or with linear pharmacokinetic. For non-linear pharmacokinetic, I think I've discussed in the previous slide. We don't have to mention again. But if for nonlinear pharmacokinetic APIs, due to saturable absorption and resulting in less than proportional increase in AUC, with increasing dose, they be conducted on at least the lowest strength. It was also in the previous slide. And for APIs with nonlinear pharmacokinetics, and the reason is limited solubility of the API and resulting in less than proportional increase in AUC with increasing dose, you need to conduct these studies with both the lowest and the highest strength. Study standardization. When you recruit your patients, I know we'll discuss about the clinical modules or the sections of the different components of bioequivalence. To minimize variability, you need to standardize exercise, diet, fluid index, posture, restrict intake of alcohol, caffeine, some certain fruit juices, and concomitant medicine. And also, when you are enrolling subjects, you need to report if there is any use of non-study medication. The factors affecting bioequivalence can be divided into two: physiological factors, that is related to the body, and then we have drug chemical properties, which is related to the drug under study. This is our whole presentation, and I just wanted to highlight it here. Now there is the discussion on the fasted or fed state. For you to decide whether we need fasted or fed state, it depends on the innovator. What does the innovator say in the SMPC? If it is fasted, you follow the fasted state. If it is fed, you follow the fed state. But what does it mean to have fasted state? The first one is dosing after overnight fast of at least 10 hours. You can give them free access to water. Prior administration of the test or the study products, no water. That is 1 hour prior administration. The volume of water to be used in the administration in 150 to 50 ml of water. And after administration, water is only permitted 2 hours and meals after 4 hours. For immediate release formulations, we need to consider products causing GI disturbances, comparator labeling requiring fed state, products with specific formulation characteristics like microemulsion, dispersion. You need both fed and fasted state, and meals. You will It is for fed state, you need to consume means 30 minutes prior to dosing. For modified release formulation, the food can influence the release of API from formulation and causing physiological changes in the GI tract. There's a term called dose damping, which is a sudden and abrupt release of API. And this is common with modified release formulation. So you need to select a meal that challenges the robustness of the MSDS to prandial effect, that is with high fat, 50%, high calorie of 800 to 1,000 kilocalories. And the patient or the subject need to start eating 30 minutes prior to administration of the product under test. That is for fed state. Sampling time. Why is it important to sample at specific timelines to avoid rejection? One of the deficiencies we observe when you are evaluating dossiers is the 15-minute time point is missing. That one allows for, like, you have so much variation in the concentration time profiles. And the sampling points should include a pre-dose sample, at least 1 to 2 points before the Cmax, 2 points around Cmax, and 3 to 4 points during the elimination phase. At least 7 sampling points. And sampling should be long enough to ensure 80% of the AUC infinity is covered, but should not be more than 72 hours. The biological fluids where we can measure our samples is blood, plasma, serum, or urine. The only problem with urine, it does not allow you to estimate the Tmax and Cmax, and storage condition detail in the analytical validation report should be strictly adhered to. The washout interval, we have said, is still be not more than 5 times the median terminal half-life of the API and minimum 7 days. But at least the washout period should allow elimination of essentially all the previous dose. There is the risk of metabolites, which are active. So extension of active metabolites with long half-life or elimination rate of API results in high variability. And you need to do assay of blood samples. Pre-dose concentration should be less than 5% of the absorbed Cmax. So the parameters to be assessed for a single dose study, we have the AUC, the Cmax and Tmax. You don't know these abbreviations are good for the audience, but the AUC is area under the curve. The concentration is, Cmax is, when you look at the concentration time profiles, the highest concentration at the peak is the Cmax, and the Tmax is is the time at which Cmax is observed is the Tmax. So area under the plasma serum blood concentration time curve from time 0 to time t is called AUC, 0 to t. You can also have AUC 0 to infinity. And Tmax is the time after administration of FPP or the product at which Cmax is observed. And T half-life is the plasma half-life, that is when half of the product is been eliminated. And the way of assessing these parameters is by using non-compartmental methods. The acceptance criteria, as we have mentioned, is 80 to 125, but for narrow therapeutic index drugs, the range is from 90 to 111. When you are doing these studies, you need to know that Cmax is more variable than AUC ratio. For statistical evaluation of Tmax is only recommended for clinically relevant claim for rapid onset action or concern about adverse effects. Comparison of median and range data for each product is adequate for Tmax evaluation assessment. Sorry. There is a component of quantification of API is by using a method, bioanalytical method. And this bioanalytical method should be fully characterized in terms of validation. And we have the parameters for validation of analytical method. I've listed them there. But during method validation, you need to have a calibration curve. And this calibration curve has a blank sample, a zero sample, and 6 to 8 non-zero samples covering the expected range. We also need to have a QC sample, which is provided at 3 different concentrations within and between redundancy and precision. There are matrix effects which need to be studied. And when using mass spectrometric methods, if you change the analytical method, you need to do partial validation. Reanalysis, reinjection, reintegration of just subject samples for reasons related to PK is not able, but if it is done, it should have been predefined in the protocol because it might lead to biased outcome. The samples from one subject, that is period 1 and period 2, should be analyzed in the same analytical run if possible. The precision and accuracy of the model should be confirmed by reanalyzing the subject sample in a separate analytical run on a different day, and that is called incurred samples reanalysis. The analytical report should have all information for calibration, the QC sample results, repeat analysis, reinjection, and reintegration, if any, and a representative number of samples, sample chromatogram. This one should be submitted to the regulator for assessment. Let's go to the next slide. Statistical analysis, we have parametric and non-parametric method. I will not talk much about it, but for parametric method, we have the ANOVAs. And non-parametric method, we have the chi-square. I don't want to talk much about it, but you can see Tmax is to be subjected to statistical analysis. You apply non-parametric methods on and transform data. You can go to the next slide. Next slide. Okay, there are special consideration, and also we have been getting questions on what do we do for a fixed dose combination. If you want to do bioequivalence for a fixed dose combination, you should compare with the pharmaceutical equivalent comparator FDC product. So if it is 2 API, you should do comparative studies with another FDC product with 2 API. But in some cases, you might have to administer free molecules, like, for example, if we have sulfamethoxazole and trimethoprim, you can administer them separate if you don't have an FDC, but the product should be in the same molar concentration. For highly variable drugs with variability of more than 30% in terms of the ANOVA coefficient of variance, you can apply a wider confidence intervals. This means you need to enroll a higher number of subjects. Scaling of bioequivalence acceptance criteria based on intra-subject standard deviation observed, Cmax is widened to a maximum of 69.84 or to 143.19. Three-way partial replicate is applied where the comparator product is administered twice, or 4-way fully replicated crossover bioequivalence study. And this is the method we use for highly variable medication. Reference scale average bioequivalence is also employed for highly variable APIs. There is another study design where we call it two-stage sequential design. This one you do an interim analysis or bioequivalence study, and then you go to the next stage. And we have two stages. The second stage is the real bioequivalence study, while the first stage is for just like pilot study. There's something called type 1 error. This should be maintained at 5%. That is a false positive, declaring a product as equivalent when they are not equivalent. I don't want to take you to too much stories of alpha and crossover and parallel study design, but two-stage sequential study can be applied to both crossover and parallel study designs. In vitro equivalence testing is like dissolution studies, and this is required for bioequivalence study, and is also for comparison purposes. As I said before, in vitro equivalence studies can be a surrogate for bioequivalence study, and especially for products which fall under BCS class 1 and 3, and also for products with additional strengths. So you don't have to conduct bioequivalence for these products. Products with narrow therapeutic index are not eligible for bioequivalence based on BCS approach. So you need to conduct 3 replicate determination of solubility at each pH condition. As I have said, 3 pH conditions of 1.2, 4.5 and 6.7 at 37 degrees centigrade in aqueous solution media. That is for high solubility, means the product is 85% of the product is soluble in 250 ml of aqueous solution or water within 15 minutes. And also we have there is very rapidly dissolving and rapidly dissolving. And for very rapidly dissolving, we have 85% in 15 minutes, and rapidly dissolving is 85% in 30 minutes. Other methods which can be employed for in vitro equivalence testing, you can use in vivo intestinal perfusion in humans, you can use in vivo or in situ intestinal perfusion using animal models, or in vitro permeation across a monolayer of cultured epithelial cells. How do we reduce risk during dissolution studies? The first one is you need to correctly classify your API. We don't want a situation whereby you classify a class 4, a BCS class 4 product as BCS class 1 to avoid conducting bioequivalence. So you need to compare the dissolution profiles, but also consider the excipients in the product. Some of the excipients can have effect on GI mortality, or might interact with the transport processes in the human body, and can even alter the PK of the API. So when you are using considering a product for in vitro dissolution, you need also to check proportionality of excipients, but there are excipients which cannot be allowed when you want to consider in vitro dissolution, that is bioequivalence. Mannitol, sorbitol surfactants, you need to assess their impacts on bioavailability of a product and should be quantitatively similar with the reference products. For class 1 API, you can be flexible with the excipients. Class 3 should be quantitatively the same and quantitatively similar to that of the reference products. As we have said, for the therapeutic index consideration before you conduct in vitro dissolution, there are those products which are bioavailable, which means they are not available at the site of action and can have reduced therapeutic effect. An example is antibiotics. But if a product is supra bioavailable, also can have toxicity issues. So dissolution measurement and the sampling time points are presented here. But one thing I want to raise here is surfactants should be avoided in dissolution testing because it kinds of accelerate the dissolution. At least you need to have 12 units for determination of profile. We can go to the next slide. There is a topic of Bioequivalence, and Bioequivalence considers the solubility and the intestinal permeability of the API. One slide which is missing, or maybe I need to give you background, is that biopharmaceutical classification classifies drug product based on solubility and also intestinal permeability. So this for one to consider for Bioequivalence, a product for Bioequivalence, you need to check the PCS classification of a product, and as I have said, class 1 and 3 are exempted. There should be also similarity of the dissolution profiles of the multi-source and comparator products at the 3 physiological pH or conditions. The excipients should be uniform or they should be similar. There is also need to consider the risk of an incorrect Bioequivalence decision in terms of therapeutic index and clinical indication for the API. So I don't want to repeat myself on the conditions and also consideration for Bioequivalence. But what I wanted to say in the last paragraph, the last bullet, is the dose solubility ratio is calculated based on the highest single therapeutic dose defined by solubility, and the volume should be less than 250 ml. That is when you can say your product is highly soluble. The products which can be considered for BCS-based Bioequivalence are immediate release dosage form, PCS class 1 APIs which are very rapidly dissolving or rapidly dissolving. And then we have PCS class 3 should be very rapidly dissolving. PCS class 2 weak acids with dose solubility ratio of less than 250 ml at the 3 physiological pHs can be considered for Bioequivalence. Drug with narrow therapeutic index is a no-go zone, no Bioequivalence. And also a regulator can come up with their criteria for Bioequivalence. So there are 3 types of Bioequivalence. As I've said already, BCS Bioequivalence. There is proportional formulation, and this is considered for products with more than one strength. So long as you have shown bioequivalence for one strength, which in most cases is the higher strength, you don't have to do bioequivalence for lower strength. And for certain dosage forms, which are listed here, you can be exempted from bioequivalence, but so long as you have implemented GMP and evidence of conformity with relevant pharmacopoeia specifications. And this is specific to oral solution. Next slide. Now we come to the Kenyan context. Where are we as a country? One of the sub-indicators of WHO was to have the same criteria applied for assessing an application regardless of their origin or destination for medical products, whether foreign, public, private, or domestic manufactured. So Kenya, we have assessed, we have conducted, or we are still in the process of conducting a feasibility study to know where we are. But what we have established so far is that we need 4 modules. And the first one is clinical, which in terms of costing, it contributes to 38% of the cost. Bioanalytical takes the highest, and this is not well established in Kenya. Pharmacokinetic and statistical analysis take 10% of the cost. And then regulatory, like in terms of being the lead in coordination and guiding the BE. This is not Regulatory Pharmacy Board. It's a CRO taking the lead in bioequivalence study, take 10% of the cost. The documentations we have or the reference I've made when I'm making my presentation is WHO guidelines, Annex 6. We have compendium of guidelines for medicines evaluation and registration in Kenya. We also have a roadmap for the implementation of bioequivalence requirement. We have a framework which is still in draft. What we require the industry or the applicant to do is to fill for us the BTIF form, which has been circulated or is available on our website. There is also Bioequivalence evaluation template. No, there are Bioequivalence application template and Bioequivalence evaluation template, and both of them there are two, one for BCS classification, BCS-based Bioequivalence and additional strength Bioequivalence. I believe this is the end of my presentation, and thank you for listening. There's been a long one. I will ask Dr. Brian to say something because of he has been evaluating with my colleagues some of the dossiers, but at least is one of the staff is with me in the bioequivalence department. So, Dr. Brian, you can say something about the deficiencies we see in applications and especially as per the BTIF form. Thank you. Dr. Brian?
I've tried to get him unmuted. He's in the room, but I'm not sure if I can unmute. But I already see there's a question. Somebody has their hand up. Probably we can give them a chance to ask the question as we get Brian up to speed. Hope that's okay, Doc.
Sorry, Dr. Brian is saying he cannot unmute himself.
Oh, okay. Let me just try get him.
Okay.
This is Kevin, I thought.
So David, do we wait for Kevin to come up before we take the questions?
Brian, not Kevin. So we're trying to get him, but you can take the questions as we get him back up to speed.
All right,
Yes.
all right. Okay. Thank you, Dr. Felicitas, for that lecture. So we'll start with Shemsu Uma. So Shemsu, I don't know if you are allowed to unmute yourself.
I've allowed her.
Okay, Shemsu, go ahead.
Okay. Hello.
Hi, we can hear you.
All right. Thank you so much. It was a nice presentation. My question is to conduct bioequivalence studies, we need to have accredited bioequivalence center, if I'm not mistaken. So what are the criteria to accredit a certain bioequivalence center? Thank you.
Dr. Felicitas, I don't know if you caught that. Maybe we can answer question one by one, or do you want me to take all of them? Okay,
I caught
great.
it.
Okay.
Yes. I did not mention that there are two approaches to having a clinical research organization. I only mentioned the consolidation of existing modules, which is the easier one for Kenya or any other country. But there is need to have a fully functional clinical research organization. The Pharmacy Board has guidelines for inspection and accreditation of clinical research organization, and they are available on our website. So what we look at is the general flow and the different components. Like the clinical component, we want to see how you recruit subjects, how you sign informed consents. There is always the approval by ethics review board. It's called institutional review boards or ethics committee. And that is the clinical component. Having also the ambulances, the beds for the patient. And then we go to the analysis. What do you have in terms of equipment? A list of equipment should be there, which will enable you conduct. And also refrigerators, you need to have them in place. When it comes to bioanalysis, you need to have the right equipment and also do the right deliver in terms of the parameters to be studied during bioequivalence studies. And then also you go to report writing. And then the overall is the quality management systems, having the right software, and also generating the right information and documentation of the whole process. But as I said, we have a clinical research organization, CRO inspection guidelines at the Pharmacy and Poisons Board.
Thank you. Thank you, Dr. Felicitas. I see a thumbs up from Shemsu, so I'm assuming that you're satisfied. We'll go then to Bina. Bina, can you unmute yourself?
Can you hear me?
Yes, we can. Thank you.
Okay, thank you, Dr. Felicitas, for this presentation. I just have 2 questions. So one was, there was a point you mentioned that before you start your bioequivalence, you need to assess the assay of your reference product, and the difference should not be more than plus or minus 5%. And I wanted to ask, so normally we get the COA from the comparator, which sometimes is very difficult. So would PPB accept if we internally analyzed the sample using our own method and then determined whether the plus minus 5% is okay? Second question, you mentioned that we have to do the dissolution in 3 media, that is the 3 pH, 1.2, 4.5, and 6.8. What if you have a 4th medium that is different from these 3, which normally we use in comparative dissolution? And then there's also, I just wanted a clarification here. There is a document that was put on the PPB portal with regard to the BCS classification, which I think has been recently put. So because you've not mentioned it, I'm not sure if it's a final document or it's something which is still subject to comments. Thank you.
I'll respond. And Brian, I don't know whether you have been able to unmute yourself. The assay, it is supposed to be done by the sponsor. And the sponsor in this case is the MH holder or the manufacturing facility. So you need to conduct, as much as you get a COA from the supplier of the comparator, you need to conduct your own assay and check the potency. If there is any deviation more than 5%, you need to reconsider the test products, the test and the reference product. But they say in the Annex 6, you can always justify, explain why there is such a difference. Number 2, the dissolution media. The minimum required is 1.2, 4.5, 6.8. If you add an additional media, there should be justification of the same. And I would not know why you will want to add a 4th media while the guidelines is saying up to the 3 physiological pHs. Maybe Pina, you can explain further. As you have mentioned, the BCS classification document, we hurriedly developed the document, I admit, because it was supposed to guide the industry in mapping of locally manufactured products. If you still feel it's inadequate, or there are some changes, amendments which can be resourceful to the industry, we can still take the document back to the stakeholders. Actually, we didn't do stakeholder consultation with this one. We've taken note. We'll review the document again.
Thank you, Dr. Felicitas. For the PH, I was just asking because there are instances where we might have a fourth one, but normally that is applicable a lot to comparative dissolution. So I just wanted to know if the same applies to BE or BE is strictly just the 3 media, because that is what is a physiological pH.
For in vitro dissolution, yes, you can add an extra media depending on your drug product, but the recommended one is the three okay. Media. So you can, if you add, you have to justify.
Okay, thank you.
Yes.
All right. Thank you. I don't know if we have any more questions. I can't see any in the queue. Oh, we actually do have a lot. We have some questions, Dr. Felicitas, on the Q&A tab. I will read maybe each and then you can answer them, quickly. All right, so we have the first question from Matovu Steven. He says, with confirmed BE of a higher strength product only, how confident may the assessor be in accepting a biowaiver application for a lower strength product? I don't know if you caught that. I'll just say it again, but feel free to also open the Q&A tab on your end. So
I have had.
Okay, great.
The question is kind of qualitative than quantitative. How confident can assessor decide on whether to give to a grant biowaiver? For our case, we have our own, as I've said, we have our own templates, evaluation templates, and also guidelines for assessment, which are internal. And when you give us a product with additional strength, we'll compare the excipients and whether there's dose proportionality. And I believe it's possible and it has been done biowaiver for additional strength. Dr. Brian, you have any answer to that?
David, were we able to get Dr. Brian to unmute?
Yes, he's able to unmute. We
Okay,
upgrade him
great.
to presenter. Yes.
Okay. Brian, take it away if you can hear.
I understand the issue of internet, but what is Stephen?
Yes, it was Stephen.
Yes, Stephen, we can always do biowaiver for lower strength. I don't know whether you're satisfied.
I think that sounds good. Stephen, if in case that's not clear, so just feel free to type again. Sangita asks, how do we go about the BE studies requirement for H. pylori kit? Are BE studies of separate molecules acceptable by PPB, or are we expected to carry out the BE studies for the kit as a whole?
Yes, in my presentation, I shared something on FDC. If you cannot get FDC, the innovator one, you can take separate products, but at least at the same dose.
Okay, thank you for
So
that.
both are acceptable, yes.
All right, so both are acceptable. Sangita, I hope that that answers your question. Then Stephen comes back and asks, thank you, Dr. Felicitas. When is the disease state considered to affect BE studies as a physiological factor, or is it completely irrelevant?
Okay, I would say most of the bioequivalence studies are conducted on healthy subjects. So there is normally an enrollment or recruitment criteria, the inclusion and exclusion criteria. So we'll go for health subjects unless now we are conducting studies which are for targeting patients, considering the safety reasons, is when we do studies on patients. But now, some patients or some subjects will have issues. Maybe they have a disease which they have not declared or they have declared. But if it doesn't affect BE, as I said also, when you are recruiting these subjects, you need to ask them about the co-administration of other medication, because at least we know it will affect also bioequivalence studies. So whatever you're saying is relevant. The disease state of the patient can affect the physiological, the outcome of the B study, that is bioavailability.
All right, thank you. I think that's quite clear. We have a question from the Africa Pharmaceutical Network. Do we have clinical research sites in Kenya that are involved in BE studies?
We have clinical research sites in Kenya, but none does bioequivalence studies. And as I've said, when we are doing feasibility study, we consider the modules which are available in the country. The only module which is lacking or inadequate is bioanalytical module. And in this case, we refer the applicants or whoever is interested in conducting bioequivalence to other countries to conduct. But okay, if for example, a CRO takes the job of doing coordinating bioequivalence study, the bioanalytical module will be conducted outside the country. And it has been happening. We have Cambridge as an example.
All right, I think that's clear. I will go to the chat. I think I saw a few questions in the chat box. From
Sorry.
Grace. Yes.
Sorry, Esther.
Sorry, yes.
Okay, when we say a clinical research organization, it doesn't mean a fully established clinical research organization. If a facility is offering any of the components of bioequivalence, it qualifies to be called a CRO. So, for example, I will use MATES. They have an analytical equipment. They can do in vitro dissolution. Cambridge can do clinical studies, that is administration of the test products and measuring the API in the blood, the body fluids we have indicated. They qualify as a CRO. So we cannot we cannot say a CRO should be fully functional and have all the components under one roof. But if it still has all the components under one roof, they are called CROs.
Okay.
I don't know whether I'm clear.
You are very clear. At
Okay.
least to me. Yeah. Okay. Grace Maina on the chats asked, what documents should one submit if an FDC is the only brand available in the market?
Please repeat the question.
What documents should one submit if an FDC is the only brand available in the market?
So if the FDC is the only brand available, do you mean the innovator or the generic or the generic products? Please, whoever asked that question, to clarify.
Okay. All right, Grace, if you don't mind clarifying on the chat, that would be nice as I move maybe to another question by generic. Sorry, she says generic.
Use a generic and it's only available in Kenyan market.
I suppose, yes. Grace, you can confirm?
Let me explain the scenarios as Grace tries to explain.
Okay.
For fixed dose combination, you look for the innovator. Because if it's a generic, there must be an innovator. And if the innovator is not available, you go for the market leader. And I gave another option. There is normally a flow chart on deciding the innovator to be used or the comparator product to be used. If the market leader is not available, the regulator can also guide after considering issues of harmonization and acceptability of the B study, not just within your own jurisdiction, but outside.
All right.
But I have said, if the FDC, you cannot get the FDC, you can use the different molecules separately. You can administer the different molecules separately.
Okay. I
Yes.
hope that answers your question, Grace. In case of anything, please feel free to raise your hand and we can unmute you to elaborate on your question. Bridget Kendi asks, in the clinical setting, she has seen various generic antihypertensives which have varied effects, if anything, producing less effect. Could this be due to lack of in vivo bioequivalence studies? What should be the way forward?
What did you say was the name of the person asking the question on clinical setting?
Bridget.
Bridget, okay.
Yes.
Bridget, please, if you can do a write-up for me, it will help me in making sure that we have bioequivalence studies taken seriously in Kenya. Because if a product is not performing well, and which of course also I've been a patient, I've taken different brands. So it's possible that we have not conducted in vivo bioequivalence studies.
All right, so I think that's possible. Okay. Thank you. I hope that answers your question, Bridget. I see a hand from Bina. Bina, so feel free to unmute as you ask your question.
Thank you. So I just want a clarification. These studies, should the comparator samples come from an ICH region or we can pick from our market? Thank you.
A comparator sample, as we have said, should come from, as we said, it is the innovator. And then if the innovator is not there, it's the market leader. And if it is not the market leader, at least you consult the regulator to give you direction on which comparator to use. But the reason why we are strict about the comparator you are using is for the credibility of your study, and also there is something called biocreep. If, for example, you use the wrong reference product for your studies, obviously they cannot be meeting the standards of efficacy, and also the parameters will be, even if it is within the normal ranges, maybe it is less. So if you study your product based on a comparator which is not well qualified, you're introducing something called biocreep, and it lessens the credibility of your study, and also we are not assured about the efficacy of bioavailability of your product, that is the efficacy.
Thank you.
All right. I don't see any other question. So I will assume that we don't have any more questions. So I think I'd like to personally take this opportunity to thank you so much, Dr. Felistas, for that lecture. It was a very informative session, and thank you also for taking your time to respond to all the questions. I really hope that everybody who was present has benefited from the lecture, and I hope that we can have you again, Dr. Felistas, for another lecture. So I think from us here at African Pharmaceutical Network, we're so grateful for all the over 100 people who joined, such a large attendance, and hope that we can keep on joining and learning together. So without further ado, I will take this opportunity to end this webinar here and to thank everybody for your time. Thank you for your attention. Thank you for your participation in this webinar. And we hope to see you again when we do host our next webinar. So good night from Kenya and good day for those who are not around East Africa. We do hope and we wish you well. Stay safe, stay healthy, and God bless. Thank you so much, Dr. Felistas. Have a good night, everybody.
Thank you. Bye-bye.
Bye.